at Northwestern in biomedical engineering. And then he and I crossed paths at Georgia Tech, where we did our PhDs together. In fact, we almost were in the same lab. So after wrapping up his PhD with Elliot Chaikoff, he actually moved on to Harvard Medical School, was a postdoc at Beth Israel for a little bit, and then did another postdoc at Rice University with Jeff Hartgerink, and is now at NJIT after publishing great papers, starting companies, and so on and so forth. So I'll let Vivek tell the story.
All right, thank you. Wait till what I say. Okay. So, morning, afternoon. Yeah, we'll go afternoon. Afternoon, everyone. My name is Vivek Kumar. As Jay mentioned, great introduction. We've known each other for quite some time. And I think what he forgot to mention was that I wouldn't be here if it weren't for Professor Sai's work and helping me, guiding me through some of those initial steps in getting a faculty job. Anyway, so today what I want to talk to you about is self-assembling peptides, which can act as biomaterials as well as drugs. That's me. We do a lot of different work in a lot of different fields, as you'll see in some of my latter slides. But I do have a couple of conflicts that I should disclose when I give any public presentation. The first of which is that some of the technologies that you will hear about today are have been patented or have intellectual protection on them, and they're being commercialized by a startup that I have called Nangiotech, right there. We made that back in 2015 while I was still in Houston with my postdoctoral PI. That company is still going strong with one employee. Anyway, the other technology that I'm going to talk about very briefly is a startup that I made for parking. Don't worry, this is actually going to be a biomaterials talk, but we'll talk about that right at the end. But again, disclosures that I have to disclose. So, I have a laboratory at NJIT, and we do a variety of different projects which utilize a platform technology, a platform capability that I'll talk to you about briefly. And these are known as self-assembling peptides. All right, so now self-assembling peptides have a basic structure, a canonical structure that looks like what you see on your top left, this guy right here. Now what I want you to do is ignore the RGDS here. I mean, clearly we all see the RGDS here, right? Ignore these amino acids right here, which help with cell adhesion. Ignore the leucine, arginine, glycine, LRG spacer here. But the main body of the peptide consists of termini with polar amino acid residues, your arginines, your lysines, as in this case, your glutamic acids, aspartic acids. So they have a charge on them. The mid-block of these self-assembling peptides, consists of alternating hydrophilic, the hydroxyl group here, as well as hydrophobic, the isobutyl group right here, of serines and leucines respectively. So we have alternating serine, leucine repeats, 6 specifically, 3 here and 3 here, that allow for self-assembly. Now I know you're on the verge of falling asleep, so I have a little animation to tell you, kind of explain how these self-assemble. But before we get to that, so one of the interesting things about these peptide materials is that we can incorporate biological functionality. And that's just a fancy way of saying, well, we did a phage display, or we looked at the literature and we found a short sequence that does the same trick as the much bigger cytokine, chemokine, growth factor, small molecule. So we take those mimics, as you can see here with RGD, which is derived from fibronectin, or an LRG, leucine, arginine, glycine sequence, which is MMP2 cleavage susceptible. So these hydrogels, these peptides in and of themselves, can potentiate cell adhesion as well as biodegrade. So we see that in aqueous solutions, the different hydrophobic regions come together. So you've got the hydrophilic region, those hydroxyls, isobutyls, the hydrophobic region, and those bioactive domains that I talked about. We can add RGD, we can add angiogenic domains, osteogenic domains, what have you. Anyway, in aqueous solution, the hydrophobic regions come together, excluding that water, right? So you can see you have lateral fiber self-assembly in the cross-perpendicular axis to those peptides. So essentially you form these multimers. As you can see here, you can do some fancy chemistry, IR, circular dichroism, to show that they form antiparallel beta sheets. But what I want you to take away from this is that that base peptide right here has a net positive charge buildup, which repels the next peptide that's coming in, right? Electrostatic like-like charge repulsion. So if we can shield those charges with these yellow balls, these phosphate ions, these multivalent anions, negative charges, we can allow charge shielding and long-range fiber self-assembly, intrapeptide, interpeptide, cross-linking, and essentially what we end up with is a nanofibrous hydrogel that you see over there. I believe that's a 1 micron scale bar. So essentially, I think the key point that I want you to take away from that previous little animation was the fact that these materials are held together by non-covalent bonds. Now, obviously, the polypeptide structure is covalently bound, but the self-assembly, the supramolecular self-assembly of these materials is guided by non-covalent interactions, right? Hydrogen bonds, van der Waals interactions, ionic interactions. And because of that, those bonds break easily and reform easily. So these materials self-assemble into hydrogels, which are thixotropic. So we can syringe aspirate them, inject them, and they re-self-assemble in situ where we inject them. Now, what we've done in the past is release a variety of different drugs, cytokines, chemokines, small molecules. This is a basic idea of what the hydrogel looks like before we add phosphate. After we add phosphate, it forms a nice little hydrogel. And again, I just want to remind you that these peptides essentially can be syringe aspirated and injected and create a depot for either cytokine or growth factor release or extended activation of specific responses. And we're going to go into some of those in a minute. So we've got a versatile platform, and one of the first things we did was say, well, can we tailor inflammation? So a couple of years ago, while I was still doing my postdoc, we said, well, can we load monocyte chemoattractant protein 1, which recruits naive monocytes towards to a region of inflammation, and can we differentiate them towards a pro-healing anti-inflammatory M2 macrophage phenotype by delivering interleukin-4, right? So we loaded MCP1 and IL-4 into these scaffolds, and essentially, and we assayed that in a relatively simple, minimally invasive rat subcutaneous model. So we take our hydrogels, syringe aspirate, lift up the skin on the back of a rat, make a little tent, and inject it in, right? We put in 200 microliters, put in whatever dose of growth factors, cytokines, or just material alone. And what's really interesting when we do histology is we see the majority of the cells that infiltrate the scaffold, right? So this is the scaffold right here behind the yellow line towards me. So unlike your traditional biomaterials, which either encapsulate, have a foreign body response, form a thick fibrous capsule layer walling off the implant, what we see is excellent host, the fascia, connective tissue here, and material interaction, where you can barely see any distinct boundary or separation between the host and the scaffold itself. However, the scaffold is chock full of cells, right? There are a ton of cells that infiltrate these scaffolds as early as 7 days, which is what you see here. Regardless of whether we add cytokines, chemokines, or what have you, majority of the cells, or a good number of cells which infiltrate these scaffolds, are CD68 positive in red, CD68 positive macrophages. So what we know, and I think I have a slide which we'll talk about in a minute, is that these macrophages infiltrate these scaffolds, gobble it up, phagocytose it, process it, and essentially biodegrade it over a 3-week or so period, laying down their own matrix. And we've done some TEM to show some of that in a minute, which I'll show you in a minute. So specific to this whole inflammation modulation story I was telling you about, if we load monocyte chemoattractant protein 1, we recruit a lot of monocytes. If we load interleukin-4, we start seeing that the interleukin-4 is being released from our scaffolds, influencing the macrophage phenotype, not just within the scaffolds, which you can barely see here in purple, stained for by CD163, an M2 macrophage marker, but also in the vicinity of the scaffold. So we see the interleukin-4 is being released from these scaffolds, tailoring the inflammatory environment in the vicinity of these hydrogels. So the same can be done for a variety of different drugs. And what's interesting is, okay, I'm going to stop there. So if you guys remember from that self-assembly video, the little yellow balls, those phosphate ions, that's pretty much all we need to formulate these materials. So you take your lyophilized peptide, dissolve it in sucrose, and then you add your PBS, and that's your full formulation. The lack of organics, the lack of any other funky solvent makes this highly biocompatible in the different environments we put it in. Now, keeping with that theme of using a multivalent anion and a negatively charged ion with multiple charges, instead of phosphate, what if we loaded a drug which has multiple negative charges? So we loaded suramin, which has multiple sulfonate charges, so you can see right here. We loaded clodronate, trypan, as well as heparin. And what we saw was that no matter what drug we load, well, as long as we loaded an anionic drug, it would hold onto that matrix. It would essentially ionically cross-link that matrix very stiffly, that's a word, and essentially release over a much longer period of time. If you remember those release charts from growth factors that you saw 2 slides ago, we saw that they come out over a 2-day to 14-day period. We see that the drug is still in there depending on loading amount all the way to about 30 days here. And we published that, again, a couple of years ago in JACS. So, the third part of this story, and I am running super fast, but that's okay. The third part of my story that I want to tell you about are functional hydrogels that we can make from these materials. Now, I told you how we can add an RGD domain. So this picture right here, we talked about inflammation modulation. You got the base peptide, you can add RGD, and essentially you can self-assemble a matrix which presents that RGD at extremely high epitope density, right? So instead of taking a whole bunch of fibronectin, a lot of growth factor, we can take short epitopes that replicate the function of the entire protein, of the entire factor, and load them onto our scaffolds. And we did that with RGD, and we showed that we had excellent in vitro as well as in vivo, in vitro cell adhesion, 2D and 3D, as well as in vivo cell infiltration. So then what we said was, can we change that RGD to something else, right? So we said, well, can we put in a bone morphogenic protein mimic, a BMP2 mimic? There are a couple of groups working on that right now. Can we put a neurogenic growth factor mimic, neurogrowth factor mimic as well? And we have a project that's going on that. Or can we put on a vascular endothelial growth factor, a VEGF mimic, a mimic which helps with the development of blood vessels? So that was pretty much the last set of work that I did during while I was finishing up my postdoc, and a number of these studies are carrying on in my independent lab for a variety of applications. But basically, I want to tell you the story of not just adding a cell adhesive sequence. So instead of adding that cell adhesive sequence RGD, we have this same base peptide scaffold which self-assembles into a beta sheet forming nanofibrous hydrogel, which is thixotropic, syringe aspirable, deliverable, stays there. Which is biodegradable, put on that MMP2 cleavage sequence, glycine spacer. We also put in a mimic of vascular endothelial growth factor, VEGF165. And that mimic, it's called QK in the literature. It was published about 12 years ago in PNAS. We tagged that mimic on to this peptide scaffold, and the exact sequence is right here. And what we saw was that when these scaffolds self-assemble, as we expected, they present this epitope at extremely high density, as you can see in this cartoon model here. And when we do in vitro, and when we probe the in vitro effect of these peptides, we see that they're actually binding to the receptors. We use human umbilical vein endothelial cells, HUVEC endothelial cells, and we show that these peptides in monomeric form or multimeric form bind to the VEGF receptor 1, VEGF receptor 2, and neuropilin 1, which are all upregulated in the presence of VEGF. So we took these injectable scaffolds. Remember, it's kind of like honey, kind of like hand sanitizer, the good quality hand sanitizer, the Purells, not the dollar store kind of. The ones that when you flip upside down, it stays there, but when you pump it, it flows. Same idea, right? Toothpaste, squeeze it, flows, toothbrush, doesn't slough off, stays there. Syringe aspirate, inject, bolus, stays there. We injected that bolus into the backs of rats, and within 7 days, and this blew us away, and blew a number of people away. But basically, within 7 days, this entire region here is scaffold. We see rapid infiltration of blood vessels. We see rapid development of blood vessels, which are not just positive for CD31 in green here, which is an endothelial cell marker. So we're forming not just capillaries, but also positive for alpha SMA, alpha smooth muscle actin, which is characteristic of smooth muscle cells. So mature arterioles, venules, and we're forming these vessels at 15 to 50 micron diameters. So these aren't just micro vessels, capillaries, nascent vessels that form quickly, that degrade quickly, that go away quickly. We're forming mature vessels, again, all within 7 days. So we're really excited about this, and you can kind of see how, at least what we hypothesize is happening. You inject this big hydrogel in there, there's an on-off, these little peptides come off, they create a chemotactic gradient to parent vasculature, which starts to infiltrate, and you can kind of see in this oblique section here, a longitudinal section of a blood vessel infiltrating the implant. So, given those exciting results, what we decided to do was to make a variety of different variants of this to make sure that what we were doing was, for lack of a better term, legit. So we made just the degradation sequence, the angiogenic sequence, as well as a fluorescently tagged sequence, because we wanted to see how these materials would be trafficked in the body. Excuse me, as well. That's that lunch burp that I was very feared about. Anyway, so we can make these injectable hydrogels. We make them fluorescent. I told you about this whole story of activating receptors. We can add peptide to cells in culture, take them off, and do flow cytometry. And if they're bright, if they show up in flow cytometry, there's a good chance our peptide was bound to them because of that specificity. And essentially what we said was to understand some of the better mechanistic questions, that whole facts story I just told you about, to understand some of the mechanistic questions, why don't we try using a mouse model? I mean, rat models are great, but you've got only that many antibodies, only that many protocols, facts, markers, or what have you So we injected this into the back of a rat. You're going to have to take my word for it. It looks slightly better on this screen. You have to take my word for it. But that's essentially my thumbnail, so you can kind of get an idea. It's about a centimeter and a half of implant right there. And you can see gross morphology of blood vessels around the implant, as well as the vascular bed that it was sitting on the flip side of a mouse, right? So we implanted a mouse. I believe this was at 3 days. We flipped it on, took the skin off, flipped it over, isolated the implant, took it out, and essentially you can see the vascular bed that was resting in, which is highly localized to the implant area, as well as microvessels visible to the eye in gross morphology. The motivation for doing that, outside of the cool picture, which you can take with your iPhone, was you can embed that whole scaffold in something called epinate. You guys remember Jurassic Park, right? Remember that amber stuff that they got the DNA out of and everything? Same idea, use extremely hard, somewhat hard resin, it's called epinate, embed your entire scaffold in there, and section it to do transmission electron microscopy. You can stain it for what you want. And what we saw was that in the middle of these implants, we have phagocytes, as you can see, this guy right here, right, blown up over here, which have invaginations taking up these filamentous peptides with the hashtag sign right there. What's interesting is that not only are cells phagocytosing this peptide scaffold, not only are they infiltrating them rapidly, but they're also depositing their own collagen. So we see the characteristic deperiodicity within collagen fibrils in the middle of these implants. So we can take these implants out and show that the cells that are infiltrating are secreting their own matrix as well. And we're showing this with histology and mesostrichrome as well as H&E and what have you. So we take out these filamentous peptides, degrade them in intracellular vacuoles, and just forget about D, and degrade them in intracellular vacuoles. So the next question is, what exactly is the cellular transport process that's going on here? Is it purely through endocytosis? Is there some transmembrane transport of the filamentous peptide into the cells? And depending on how this material is presented to the cell, potentially we can modify these peptides, which rapidly infiltrate with APCs, CD68 positive macrophages, which are APCs, antigen presenting cells, process antigen tacked onto our peptide, and present them at extremely high density on MHC, right? So class 1 or class 2. For those of you who didn't understand any of that, we can try to make vaccines with these guys. So anyway, we did a very quick facts analysis. This is rough data, relatively small numbers. But essentially what we see is that majority of the cells that start infiltrating these implants, at least at early stages, are macrophages, your F480 marker right here. We see that, well, rather, almost all the cells which infiltrate these scaffolds are CD45 positive hematopoietic cells. I know that's a relatively small number, still going through controls, but anyway, so in this initial study, what we saw was that majority of the cells that infiltrate are not humoral, are not cells of the humoral response system, so they're not B cells or T cells compared to tissue, CD19 for B cells as well as CD3E for T cells. However, we did see an uptick in T cells that we further characterized to be NK1.1 cells, so not so much your classical CD4, CD8 T cells associated with your humoral response, but more your innate NK cells, which try to figure out, hey, what's going on? Let's get rid of this if it's foreign, or let's work with it if it's bioactive. So, okay, that didn't work. Maybe that did. Okay, so remember that whole fluorescent peptide we made? We said, well, if we made it, might as well use it. So we injected it into the back. No, sorry, I take that back. If we're doing animal studies, if we made it, we're going to find a defined study to use it in. So we took mice and we injected 200 microliters of fluorescent peptide, fluorescent hydrogel, into the dorsal subcutaneous aspect, right? So into the backs. And we saw the material degraded over about 10 to 12 day period. Now keep in mind, when we sacrifice these animals, flip the skin over, the material's still there. You just can't see it that well because of autofluorescence of skin, urine, which is sometimes on the skin, hair, as well as the skin itself has a certain attenuation of fluorescent signal. So what we would like to do is to actually carry the study on into something more defined, maybe use radioisotope labeling to understand pharmacokinetics and pharmacodynamics. Anyway, long story short, we can inject the peptide into the backs of mice, and we see that they degrade over about a 2-week period. Similarly, skipping from A to C, similarly, if we inject it in the intramuscular space, so this was A was a 200 microliter injection, this was a 50 microliter injection, we see that the material actually sticks around for a little while longer. By day 12, we see a considerable amount of the material still there. And one of the hypotheses that we had was, well, the skin's thinner here, there's less hair there, so that's one reason. But in addition to that, there's a lot less interstitial flow. So the tissue flow in the subcutaneous space or what have you is a lot greater than in the intramuscular space. You just don't have as much flow going into there. So then we said, well, what happens if when we're injecting these injectable materials, instead of going into the muscle or under the skin or what have you, we nick a blood vessel and we inject directly into the blood vessel? What happens then, right? Do we get a thromboembolic event? Do we have blood clotting on the material, stroking different tissues or organs? Or do we see what we saw here, which is because these materials are shear thinning, they're thixotropic, when they feel a force, they flow. When they feel hemodynamic shear, blood flow, in the tail vein where we injected it, almost immediately, so actually really within 10 minutes, it localizes to the urinary bladder, so it gets cleared by the kidneys, gets localized in urinary bladder, where there's relatively no flow until the animal urinates. And what we see is that the material is localized to the bladder all the way out to about 2 weeks as well. So anyway, so we're really excited about this, and this was a lot of this work was done at Rice University while I was still a postdoc, and we worked with the Texas Heart Institute, some of the folks over there, to see whether this had any translational potential. Can we take this technology and move this towards the clinic? So we said, well, what is the first model we should try? Well, why don't we look at the heart? Why don't we go straight to the heart where the money is, right? $32 billion market. Let's develop a material which helps regenerate cardiac tissue. Now, there were multiple reasons why that didn't happen, against my will. And primarily because if you think about therapeutics for myocardial regeneration, heart muscle regeneration, there is to date no FDA-approved therapy, right? No therapy which is approved for regeneration of heart muscle. And that's primarily because your phase 2 clinical endpoints for efficacy and your phase 3 clinical endpoints for widespread use and widespread efficacy is extremely difficult to do when your endpoint is death, right? Does the patient die? No? All right, well, you have better efficacy with your material. The other factor is typically when a person has a myocardial infarction, has a heart attack, they have a lot of comorbidities going on. They have a lot, and the environment is much more complicated than just simple angiogenesis, as Professor Sai would tell you in some of the work that he's done. So we decided to go with a lower-hanging $11 billion indication called peripheral artery disease. So imagine heart attack in your legs, right? So if you have poor circulation in your legs, not in the macrovasculature, not in the big vessels, which are 4 to 6 millimeters up to a centimeter in diameter, but in the microvessels, blood vessels which are on the order of 100, less than 100 microns, up to about 200, 250 microns. Our hypothesis was if you can deliver an angiogenic stimulus directly into the gastroc, into the calf, perhaps you can regenerate blood vessels to better perfuse the tissue. Before you think I'm crazy, they've been doing this for about 30 years with stem cells, gene therapies, growth factor therapy, and they've worked so hard to purify enough growth factor, spends hundreds of thousands of dollars for these studies, for the studies, millions, tens of millions of dollars, but for individual injections, only to realize you shoot it in, within a day or two, it's gone, right? It diffuses away. So what if you had a matrix to hold on to that? Secondarily, what if you had a matrix that was intrinsically signaling the cells, right? What if you had a scaffold that self-assembles into a hydrogel that had that receptor mimic on it that intrinsically activated cellular phenotype? So anyway, we did an animal efficacy study, the study that you do right before going to phase 1, clinical trials. Of course, you got to do your tox studies and your manufacturing studies. But anyway, so we did an animal efficacy study. We ligated the femoral artery. We tied up the artery that supplies the leg with blood. We injected our material in the quadricep and calf, about 25 microliters each. We did laser Doppler perfusion imaging. So here's where I take off one cap, put on the other cap. I can legally say these things. No. So I have a conflict of interest. So the startup that I have is trying to commercialize this technology. So FYI, that's why this little blanked out part right here. So I'm going to talk one slide on the work that I'm doing there. So we did our animal efficacy study. We tied up the femoral artery. You can see in laser Doppler perfusion imaging, ultrasound with color, right? Tells you blood flow. We can see with color blood flow essentially with ultrasound. Good perfusion is in red, bad perfusion is in blue. You tied up a big blood vessel. You're going to have pretty poor perfusion. Inject the material. Within 7 days, we see restoration of perfusion, not just past the ligature site where we tied it off, right? Good perfusion there, but also in the foot pad. And this is important for those of us who are concerned more about diabetic foot ulcers or diabetic wound healing. You want to increase vasculature throughout the limb and not just in one area. But anyway, so coming back to the point, we saw that progression and improvement in perfusion persisted all the way out to about 28 days in the old mice that we have, in both the adult and old mice that we had. So basically we quantified that, and we saw that after 30 days and 8-month-old mice, this is actually 13-month-old mice data. So after 30 days and 13-month-old mice, what we saw was that we had slightly better perfusion of the tissue, close to 90-95% recovery of perfusion, while the base peptide without that angiogenic domain, or just the carrier, saline, had virtually had very minimal effect. We do see we see a decent amount of regeneration because a lot of these animals are highly auto-regenerative. They heal their wounds relatively well. They've evolved for millions of years to do that, which is partly why we use adult mice, 8-month-old mice, as well as what I like to call geriatric mice, 13-month-old mice. And we, good, got some laughs there. And we let them go out to 3 months each, and we see that continuous uptick in perfusion. Now, in addition to that, so if you think about, well, you can't really, I guess you could do imaging studies in humans as well, but tracking flow of microvasculature in the calf is relatively difficult to do. So the FDA's mandated phase 2 clinical endpoint, how do you show success in phase 2 clinical trials, is essentially an improvement in treadmill endurance, right? So not looking at death as an endpoint, we're looking at improved treadmill endurance, right? So the guy gets on a gets on a treadmill for 5 minutes, how far did you walk, come back in a month, come back in a couple of weeks, how far do you walk, and you keep doing that for 3 to 6 months. And what we saw was that before surgery, these mice were running about 450 meters or so, and after surgery and injection of our material 21 days later, we see that mice were running just as far, about 450, slightly more than that, compared to controls. So we showed efficacy not just in terms, we showed a mechanism in terms of receptor activation in vitro, and we also showed a mechanism for efficacy, and we demonstrated efficacy. So we took that technology. I started a startup that I'm the CEO of, I believe. So many conflicts. And essentially, we're trying to do fundraising, right? So a part of my job is to explain all this stuff and a lot of the other stuff to average everyday people. So it's been, as a side note, it's been quite an interesting experience. So if anyone's interested in innovation and pitching and putting together your research towards commercialization, SBIR, STTR, sit on SBIR, STTR panels, feel free to reach out. Anyway, so we have a startup that's trying to take that towards phase 1 clinical trials right now. I mentioned that one employee that we have. He's one of the co-founders, and essentially he's working for equity, which is what that means. And he's writing up some SBIR, STTR grants to try to move this forward. And if any of you do go down this path, what you'll realize is that every time you go to an investor, an angel investor, small time, a venture capital investor, bigger time, private equity, you got a phone call, great. When you go to them, they'll keep telling you, come back when you have a little bit more data, come back when you're in human, come back when you're in phase 2, come back when you have a product on the market. And quite honestly, by that time, you don't need their money. But okay, so that's a little bit of the stuff that was taken out of the lab towards the clinic, or at least trying to take it towards the clinic. What we're still doing in the lab is saying, well, we've done this whole angiogenic story, or at least we've got some traction on that. Let's see where that takes us. What did we put on? What if we use that same platform technology? There are some modifications, which I'm not going to go into, but use that same base peptide, which self-assembles into a hydrogel. And instead of putting on an angiogenic domain that helps blood vessels form, right, angiogenic, let's put on a dentinogenic domain, or let's put on a neurogenic domain, like Patricia's working on. Let's use these materials as cell carriers. I mentioned that thixotropic cells like them, and basically you can resuspend your cell pellet within these hydrogels, inject them with a 30-gauge needle. The smaller the needle, higher the shear, better it flows, right? So things to think about. So you can use them to deliver a variety of different cells. Specifically, Tony's working on delivering cardiomyocytes. We can incorporate a variety of different types of these peptides, anti-inflammatory peptides, pro-angiogenic peptides to decrease neointimal hyperplasia and increase vascular vasorum formation. For any of you know about vascular grafts, we're trying to do that as well. And in fact, the work on dental pulp regeneration as well as vascular grafts we presented at BMES last week. I'm going to skip cupping for a minute. I'm going to very briefly go through these. So if we can do pro-angiogenic therapies, why can't we do anti-angiogenic therapies? So we looked for a domain from a tissue plasminogen, something that breaks up clots, something that disrupts endothelial cell function, and we found one. We tacked on our peptides. We're trying to start some rapid intraocular diabetic retinopathy studies. Basically, a bunch of blood vessels on the retina, injected in the eye, see if we can get rid of those, all right? Now, yeah, let me just go on that. So and the other thing I mentioned just now was that these materials rapidly infiltrate with CD68 positive macrophages, which are antigen presenting cells. So we're trying to take the V2 loop from HIV's protein coat, in collaboration with Abraham Pinter at the Rutgers School of Public Health, attaching it to our peptides, with the rationale that if we can present an antigen at extremely high density in a self-adjuvanting material, in a material that draws those immune cells in, presents it nicely presents the antigen, the immunogen, thing that you're trying to vaccinate against, nicely in high density to the antigen presenting cell, hopefully you can stimulate better antibody responses. I mentioned lysine just now. If you increase the number of lysines, like your canned soup, right? It has polyl lysine in it, or other preservatives as well. Hopefully, with increasing the number of lysines, we can make these materials bactericidal. So we're taking a look at that with E. coli, I believe. And we're looking at a couple of other things as well, including drug delivery, degradation to understand degradation profiles, and a little bit of work on Alzheimer's because they form amyloid beta plaque and we form amyloid beta plaque. I'm not going to discuss it in detail because. So the last thing, so I want to very quickly talk about some of the data we've started to generate on dental pulp regeneration, just as a case study on the general approach that we're taking to these materials, at least from the bench side towards translation. But before that, I want to tell you very briefly about what Cheryl does. So I got into this whole world of peripheral artery disease, right? Poor circulation in the legs, and I'm thinking to myself, okay, well, we had these crazy peptides we've got, which are essentially drugs. I think. I might have failed to mention that. I'll come back to it in a second. These materials are classified as drugs by the FDA. I can tell you why in a second. But I said, well, what is an easier way for me to get a little bit of that clinical experience exposure, clinical trial experience exposure? Because it really helps when you go talk to VCs, when you go talk to investors, or even when you're writing federal grants, to have an idea of that end goal. So what I said was, well, what is a relatively non-invasive, simple study to do that we can get some interesting data on, right? So if you guys remember, I know this seems a little wacky, but if you guys remember the Olympics last year, right? 2016 last year? Yeah, about it. Last year-ish. You guys remember Michael Phelps' back? Remember all those cup marks on his back? Right? So essentially the sports industry field has realized that using cupping, this ancient Chinese or Indian, whatever technique of heating up a cup, creating a vacuum, putting that vacuum on the skin, which raises the skin, causing microvascular damage in the dermis, results in better muscle healing. And I can't say the word that I thought. I didn't quite agree with that. I didn't quite think that would work. But essentially what we're trying to do is to use cupping to create microvascular damage on the calf and seeing whether that increased tissue flow to heal up the hematoma can improve muscle, can stimulate angiogenesis and improve muscle function, right? So essentially what Cheryl is doing, she's written up an IRB. We're running a pseudo clinical trial. We're developing preliminary data to submit a clinical trial planning grant in R34, hopefully next year. So we've got a couple of kids coming in, running on the track. I'm going to cup them, bring them back next week, and see whether they have any exercise improvement. Anyway, so that used to say anger management for the BMES. I figured. Anyway, so let me tell you very quickly about the dental pulp regeneration that we're doing. The reason we're doing dental pulp regeneration was because after I did my PhD at Georgia Tech, after I was at Beth Israel Deaconess, I was at Rice University for a little while, and I worked for a chemist who had a dentist as a collaborator. And when you work for a chemist or any kind of material scientist, in my opinion, you make a material and you throw it at the wall, see what sticks, right? So we realized that these materials might be helpful in dental applications, like filling up the pulp after you have infection, to take out the diseased pulp, capping it, and allowing new pulp to regenerate instead of putting in rubber rods, okay? So essentially we're working, so I carried on some of that work, and we're working with a clinical a dental collaborator to implant some of these scaffolds. And I'll explain that story in a minute, but what I did want to touch on is the fact that these materials are drugs. They're made by solid phase peptide synthesis. They're not made by expression, so they're not expressed in E. coli or yeast or what have you. We have a machine that tacks on, like Lego blocks, right? Amino acid by amino acid. They're made by solid phase peptide synthesis, and because of that, they're not a biologic, and they are a drug or a device. If they form a scaffold and don't really interact with the body otherwise, they'd be a device. But if you have a domain that specifically interacts with very specific cells through a mechanism that you claim is beneficial for the efficacy of the drug, long story short, if that stuff sticks to a cell and does what you ask it to, you essentially form drugs. All right. So these materials, I tried to weasel in a device with the FDA. I tried to ask them if they'd give me biologic, but I think they pretty firmly told me it's a drug and don't try it. So anyway, so we took that base peptide scaffold that self-assembles into a hydrogel. We tacked on a domain, a mimic called dentonin. It is a matrix extracellular phosphoglycoprotein mimic. MEEP is a sibling family of protein, which is a small integrin binding ligand end-linked glycoprotein found in bone and dental tissues. Long story short, that little guy over there, dentonin, potentiates dental pulp stem cell proliferation. The stem cells in your pulp, it stimulates them to proliferate. We tacked it on, and we synthesized the material. We did SEM, and I love this picture, which is why it's on the whole screen. But essentially, we see this beautiful nanofibrous structure, right, that forms. It's a 1 micron scale bar right here that forms in aqueous solution. Now, keep in mind, we can syringe aspirate these hydrogels and inject them into different cavities. So we looked at the rheological properties. They form hydrogels. It's a little more not as daring as I would have done. I would have flipped it all the way upside down. Now, it forms pretty good hydrogels. We've done some chemistry, some IR to show they form antiparallel beta sheets. We've done some rheology to show that it forms indeed these thixotropic materials. Just to very quickly explain rheology in less than a minute. Your storage modulus, your G prime, the triangles over there are how solid your material is. The G double prime, the loss modulus, is how liquidy your materials are. And they exhibit both liquid, solid properties and liquid properties. They're viscoelastic. And when that G double prime intersects with your G prime, either when you're turning very quickly or shearing it at a high shear rate, you'll see an inversion where those liquid properties mask or eclipse the solid properties. And essentially, you have a syringe aspirable, you have a liquid, right? So at high angular frequencies, at high strain rates, as well as at high amounts of strain, close to 70 or 80% strain and close to about 70 or 80 radians per second angular frequency, we see that these materials liquefy, right? Then the question is, how long do they take before they come back to their original mechanical properties? So essentially, we did a shear recovery experiment where we started out these hydrogels at a relatively low oscillatory shear stress, right? So this is at 1 radians per second, which is where it's still a gel, not really a liquid, and for about 30 minutes. Then for 90 seconds, we switched that to 100 radians per second, angular frequency of 100 radians per second, and we see that immediately the gel liquefies. So this is a G prime, it rapidly drops. But within 90 seconds, and then we release it after 90 seconds, and we see that in another 60 to 90 seconds that we recover close to 90 to 95% of that G prime. I'm going to get as steady as I can. You see what I mean, right? So after we inject it, it re-self-assembles, and you regain that mechanical strength very, very quickly. So what we did, perfect. Okay, what we did was we also did AFM, so atomic form scanning electron microscopy gives you these pretty pictures, gives you an idea of the morphology, more topographical information. What we did was to throw in our peptide nanofibers at relatively low concentration onto cleaved mica and did AFM on them. And what we see is that the width of these peptides is right around 25 to 30 nanometers, and the height of these peptides is about 2 nanometers. So essentially, we have these little ribbons that are forming, which correspond nicely to their molecular, to the expected length or expected dimensions in molecular modeling. So we know that these form nice nanofibrous hydrogels. We've done some cytocompatibility. Unfortunately, I was flying. I wasn't able to get a better live dead stain before I gave this talk, but that's what we have right now. And they had, long story short, they're relatively cytocompatible. And when we inject them, we're able to inject them at a variety of different concentrations, pHs, or what have you. And some of the initial injections that we did, we put in pretty much, we put in low concentration peptide, the peptide you saw just now. And what we saw was that within 7 days, the material had completely degraded and resorbed into the system, right? So now what we're trying to do is to increase the concentration and get the pH to right about 7, 7.4, and inject it in the subcutaneous space. So again, the overall idea of what we're trying to do is we're trying to the model at least that we're going to try to do, we have an ICO for it, so hopefully this gets done soon. We're going to take off some of the enamel, the white stuff on the outside, the hard stuff, take out some of the dentin, which is the spongy stuff right below that, and take out the pulp, right? Do a pulpectomy, the soft tissue inside. Now the soft tissue inside, that's what's responsible for sensation, innervation, preservation of the tooth. Typically what they do when there's an infection here, they go in there, rip it all out, and put in rubber rods and seal it. And your tooth is prone to reinfection, it's not responsive anymore, it's prone to dropping out, falling out, and bone resorption. Alveolar bone resorption right here. But anyway, so we can take out the pulp, we can inject our hydrogel materials, and the idea is hopefully in this dog model that we're going to try to do, we have regeneration of vital pulp in that tissue, which again is responsive to external stimuli. So using that same idea, specific to that, we're working with Saul Wiener as well as Emi Shimizu over at RSDM. But using that same idea, taking these injectable scaffolds, we're working with the chief of vascular, Michael Curie, as well as one of the other surgeons there, Joe Huang, and looking at how we can improve the compatibility, or rather, improve the biocompatibility of vascular grafts. And we have a aortofemoral, aortoiliac model that we're trying to develop. So basically a bypass graft from the aorta to the iliac artery. In addition to that, I talked to you a little bit about the immunization work that I'm doing, that we're trying to do with the Rutgers School of Public Health. And then, of course, all these guys are around us. I figure, get them geographically located around NJIT, where I'm working with James Harawa on fluid percussion injury. So using our same peptide-based scaffolds after an injury, inject your material, see whether you get better regeneration. Cristiano Diaz to model some of these materials. I'm no modeler, so we're going to let him do that. I've got some. I'm working with Alice Lee. I'm going to skip Sagnick for a second. I'm working with Alice Lee to look at delivery of embryonic stem cells. She works with a number of different cell lines, as well as Hans Chowdhury on the cupping stuff I told you about a little while ago. And Sagnick is actually some of the stuff that I do outside, well, somewhat outside of my lab. So I was having lunch with Professor Sai, and he was telling me, he's mentoring a capstone team, and that I was like, oh, definitely mention that capstone team that I'm mentoring. So what I said was, so one of the big problems in New York, right? Newark, right next to New York, so we have a lot of the same problems without the traffic. And I'll get to that in a minute. But, right? So basically, one of the biggest problems with sexually transmitted diseases, especially in places like the Bronx, places that are highly diverse but just don't have the same level of protection, especially amongst men's male sexual contacts with male, right? MSMs. The rate of STD transmission is extremely high. So what we said was, what is the best way to get people to take STD tests or do STD tests quickly, discreetly, and or even give that capability to a doctor? Because one of the biggest problems out there is that the patient comes in and they say, well, I had unprotected intercourse with so-and-so, get me on prophylaxis. Or my boyfriend or girlfriend or what have you has HIV or some other disease. Can you put me on prophylaxis before we consummate the relationship? So what we said was, well, what if we developed an STD testing device? Exists, CVS, $25. You test one STD. What if we developed an STD testing device that had high specificity, high sensitivity, fewer false positives, and got the right disease at the right time, but tested for 3 STDs: gonorrhea, chlamydia, trichomoniasis, and which develops in less than a minute, right? And which costs less than $1. The overall idea is, can we develop a test which rapidly screens for STDs? So you're in the heat of the moment, you go home, you're like, hey, do you mind using the bathroom real quick? Right? So we're trying to have the analyte be urine. And at the same time, this might be a good test that we could put in doctors' clinics and PCP's clinics, so that when they see these patients, they don't need to say, hey, let me run the PCR test, let me run this test in the lab, and then start you on prophylaxis, or then start you on antibiotics, or then start you on something. And the likelihood of them coming back to the clinic is extremely low. So to improve patient compliance, as well as to reduce the overall spread. So those are the different scientific projects that we're working on. But there's another little piece of this puzzle that I didn't mention. When I first moved from Houston, beautiful city, tons of parking, not have to worry about anything. Focus on parking. I moved from Houston. I told myself, why would I live in Newark, of all places, right? Let me live in the Big Apple. Let me live right smack in the middle of Manhattan, right? So I moved to 98th and, Columbus? Yeah, 98th and Broadway. 98th and Broadway. West 98th and Broadway. And the thing is, driving into work, no problem. Just take G-dubs when it's not close by, Christy. Just take G-dubs all the way over. It takes me about 25 minutes. Park, go to work, finish, go home, spend over an hour looking for a parking spot, right? Because there's parking lots where you pay, I don't know, where you pay about $50 to $70 a night, or between $450, which is the cheapest I found, all the way to $700 a month, right, for a dedicated parking spot. But if you're lucky, you can get alternate side street parking. For those of you who know what that means, good. You can get free parking, right, if you spend enough time looking for a spot. So I spent on the order of 30 minutes to an hour looking for a spot. And I told myself, this cannot happen. So I moved to Jersey. I moved to Newark. But I also decided, oh, there's no audio. Oh, God. Okay, well, we decided to make an app called Pull Up, and I'll just walk you through it. Basically, we said, let's make Uber for parking, right? And it's called Pull Up. You can post your parking spot. You can tell the person who's coming in how long you're willing to wait, how much you want to charge for that, the time you're willing to wait, all this IP there. And essentially, we're about to release that app on the iStore soon. Now, the reason I bring that up is more of this basic idea of we're engineers, right? And I just started this whole engineering professor thing at NJIT. We're trying to develop new technologies. But fundamentally, we look for problems and we find solutions to them, be it in any of these things or in average everyday life problems. So you don't need to be a coder. I'm sorry, I didn't mean to point myself. You don't need to be a coder. You don't need to be biomaterial science. You don't have to have all the expertise, but as long as you know how to bring those teams together and motivate them, you get a lot of work done. Ah, there we go. Yeah, that's me right there. And have a lot of anger. Okay. All right. So I think that's pretty much it. I'm gonna stop there. I think I'm in time. Yeah. If you have any questions, I'd love to take them.
Do you have any questions for Professor Kumar?
Oh, come on. Yes. Yes. Oh, sorry.
Right.
So, with your mouse PED study,
Mm-hmm.
what levels of necrosis or tissue damage did you see after you ligated the femoral artery, and how was that alleviated through the angiogenesis that occurred?
Yeah. That's a solid question. You write that down. I would. So we didn't specifically look for reactive oxygen species, tissue necrosis, or what have you. We looked at more of a clinical functional outcome. That being said, these studies have been done for decades. They're highly benchmarked. We just took a model that was already established to demonstrating efficacy and move it forward. So yeah, I guess that answers your question. We didn't specifically look for apoptosis, tissue necrosis, or what have you. But in our primary indicator of perfusion, that's what we saw.
Follow up on this.
Yeah.
Do you need all of your peptides to have that mimetic, or can you dilute it out with non?
That's a great. Oh my God. So here's the thing, right? We're making this dental scaffold, and the hypothesis there was: can we regenerate? Can we get dental pulp stem cells? Some cells in that niche may or may not be there to grow into the scaffold. Secondly, we want angiogenesis too. We want to make sure that scaffold, that new tissue, is well perfused. Maybe we want innervation as well. So the question is, can we, instead of putting an angiogenic domain only, can we have maybe 30% of this guy, 30% of a neurogenic domain, and 30% of dentinogenic domain? It seems extremely easy. We just haven't done it yet. Yeah, which is real cop out, but yeah. I mean, actually, to be fair, the reason we haven't done it yet is we're doing all of this, so these guys just haven't had the bandwidth, and they're trying, but it's only that angry I can get.
So how much does that side group actually affect self-assembly?
So that's the other problem, right? So there have been I'm not the first person to think of this strategy. There have been a couple of folks before me. In fact, there were a couple of folks out there that were doing this as well. Most of the people out there either use a similar self-assembly scaffold, be it an aliphatic chain, what have you, with Stoops self-assembling peptides, or a polypeptide backbone like this, like Puramatrix Rada out of Zhang's group at MIT. But I think the real kicker is, what do you put on at the end, right? You've got the self-assembling domain. With solid-phase peptide synthesis, you can usually make peptides that are 15 to 50 amino acids long before your yields just plummet. So if we put on a mimic that is a beta barrel that is hundreds, thousands of amino acids, maybe even made by expression, and then have a short chain that self-assembles, we shouldn't expect, and I've seen a couple of papers out of Joe Collier's group that does this, but we shouldn't expect to see good hydrogellation formation, right? You still might see some of those multimers and macromers form in your best TEM picture, but the problem is, if you put on a huge sterically hindering domain, it significantly interferes with self-assembly. All of that said, depending on the self-assembling domain, you might actually complement some of the structural formation. So if you have alternating repeats of amino acids, specific amino acids in your mimic even, you might actually potentiate self-assembly. So it's a little bit of a black box. It's a little bit of understanding peptide design, neither of which I don't think anybody knows, but ultimately it's empirical. And one of the biggest challenges that these guys have had is trying to figure out how to dissolve these in the right buffer, in the right solutions or what have you. Yeah?
Following up with that, you said that these were charge stabilized, is that correct?
Mm-hmm.
Do you have any problems with salt in the mouth or actually losing their ability to stay self-assembled? Not only that, do you check the self-assembly or stability of your system in increasing concentrations of something like albumin, which might compete with the self-assembly process or even stability?
Right. So let me answer your question moving backwards. So when we load growth factors, that very first story about MCP1, interleukin-4, and what have you, every time we load growth factors, cytokines, small molecule drugs, what have you, when we buy them from R&D, scientific, we ask them for carrier-free growth factors. And for everybody who doesn't know what carrier is, I didn't know either up until a couple of years ago. It basically means without what is this, BSA, right? Or some kind of albumin to stabilize the protein. In the same way, maybe not in an identical way, but if we add BSA, if we add FBS or what have you to these peptides in cell culture, they chunk out, they precipitate out, right? Because they essentially bind to it. So typically we resuspend cells in serum-free media and then put drip-full media on top. So working backwards. So if we were to change the ionic strength of the peptides, work that's been published from the Hartgren group close to a decade ago now, has shown that within the physiologic range of salt, sodium chloride, phosphates, and what have you, these hydrogels are very stable. They form a beautiful hydrogel, but when you get to 100 millimolar phosphate or even higher, you just start seeing things crash out because you've got so much ionic strength there that nothing's really self-assembling. To just finish up the point, the primary thing that's potentiating self-assembly, at least in our opinion, even in DI water, you see hydrogels form, is hydrophobic domain, right? The alternating hydrophilic, hydrophobic amino acids, along with that charge domain. Because even in pure water, you're still going to have a little bit of ions. When you pH it, you're going to have a little bit of acetate, hydrogen chloride, HCl, or what have you, in there. So we still form hydrogels. The idea of charge shielding is to allow that long-range fiber self-assembly. So when we do TEM or AFM, after adding calcium, after adding the corresponding multivalent salts, so if it's positive charge, negative charge, positive charge, you'll see a much better network of fibers form in AFM as well. So they're relatively agnostic to charge, but within reason. Yeah. So to carry that thought, collagen mimetic peptides, a lot of collagen mimetic peptides out there. A little bit of work I've worked on briefly use charge stabilization in salt bridges, but these are they're more stable than those specific interactions required for self-assembly. Okay. Yes.
Can you get different shapes of self-assembly, like nanoparticles?
So to be fair, I don't think we've tried in. Okay, let's stick with this one. I don't think we've really tried in earnest to form other supramolecular species with these outside of just hydrogels. Now, what we do know, I'll answer the question that I do know the answer to. What we do know is that we can load nanoparticles, microparticles, liposomes into these, and they coexist. So if you think about it, these materials really boil it down to soap, right? It's a surfactant, has multiple charges, is amphiphilic, and they should disrupt liposomes. But what we see is that, and we published a paper on this, releasing multiple growth factors from the media as well as liposomes at differential rates, is that the liposomes coexist with the nanofibrous scaffold. one of the things that we like about this technology is that it conforms, right? It conforms to the shape of the cavity that you put it in. And this is something that I'm very interested in collaborating with Jay on to look at whether some of his models can be useful. For example, loading drugs into my hydrogels and injecting them into some of his models to see whether we can hit margins for tumors or better activate specific responses in the localized in locale of tissues. So I think taking all of this key points, right? Key points: injectable scaffold, self-assembling scaffold. Sorry, I'm just scrolling through this. Injectable scaffold, self-assembling scaffold can be modified to make a drug, right? Intrinsically, they're drugs. Put a different mimic on. Can support tissue, excellent tissue infiltration and growth. Yeah. So if you guys want more information on this technology or anything like that, you can they reach out to you? Yes, sure. They can reach out to Professor Sai. In addition to that, my email address is VAK, right? VAK, my initials, at njit.edu, or my lab, Kumar Lab, my last name KUMAR, lab.njit.edu. And on that lab website, I implore you to check out the other stuff we do, including all of this stuff. Just random projects that we're trying to achieve.
With no further questions, please let me thank Dr. Kumar.
Thank you. All right. Thank you very much. Oh, good timing. Thank you. Thanks for having me. Yeah, I think we've got a couple of minutes. Jay, did you want to come with, or so I'm trying to meet with?
Listen, I also want to introduce Adam. Yeah.